Hi, my name is Klaske van Wijngaarden and I am a PhD student working at the BIFoR FACE facility. I am gonna tell you a bit more about my research in the coming lecture and how it helps us better understand how trees deal with the elevated carbon dioxide levels in the atmosphere and what impact it has on their woody tissues. First, I would like to explain a bit more on what the FACE facility actually is and looks like. So FACE stands for Free Air Carbon Enrichment, which basically means that in this experimental setup, extra carbon dioxide is pumped into the air. And because of that, we can see how a complete forest would respond to that change. In order to make that work, we would also need a control environment where everything's the same except the additional carbon dioxide, because that is our treatment in this case. So some of the towers that you see in these two pictures pump normal air into the forest. So we can be more certain that the changes that we would see in this forest would be happening because of that additional carbon dioxide. On the left-hand side, you see BIFoR FACE, and on the right-hand side, you see EucFACE. Both of them are FACE facilities that I am actually working at. The additional carbon dioxide that we expect to see in like 30 or 40 years is the amount extra that we pump into the air in this experiment. So we like to call it a climate change time machine, because we look at how a forest will respond to something that will be happening in the future. So when I say I am looking at how trees deal with elevated carbon dioxide that we will see in the future, it might be good to do a recap first on how trees use carbon dioxide in normal circumstances. So using sunlight, the available water, and of course the CO2, the carbon dioxide, trees create glucose and oxygen. And that glucose is used for building basically everything in the trees, like more leaves, flowers, fruits, the root system, and also wood. And wood will support the weight of all these other components that I just summed up and will cause trees to grow taller. And it locks that carbon that is absorbed out of the atmosphere in wood, because it tends to live longer in trees than, say, leaves that will fall off at one point during winter, etc. So it is really interesting to know why or how trees change under elevated CO2, and if that also happens with the woody tissue that stores or locks away that carbon for a certain time, because it's relevant to know how long that would stay out of the atmosphere. And that is why I'm researching the change in woody tissues under elevated CO2. So, yeah, repeating, why is this important? Forests make up a big portion of how much carbon is absorbed from the atmosphere on a global scale, and it is still quite uncertain how much these forests help store away that carbon for a prolonged period of time. To look at woody tissue and growth in a complete forest is quite a big task, and I cannot manually measure all the different branches that are at BIFoR FACE and EucFACE by myself. That's why I used a scanner to make a digital copy of all the trees in these research plots. After I made the scans, I filtered away the unnecessary points that were too far out of the treatment effect zone. I separated the oak trees from the understory trees and the infrastructure. And what I end up with are single 3-point clouds. And these single 3-point clouds I can divvy up in different cylinders that will fit into the form of a tree, as you can see on the most right-hand figure. And these cylinders help me better put a number on the amount of carbon that would be stored in these woody tissues. And in order to show a bit on what a point cloud like that looks like, I made a video that you can see in a bit to experience what walking through a digital copy of BIFoR FACE would look like. Here we are walking through the digital copy of a BIFoR FACE forest plot, showing the points that reflect the understory, the infrastructure as well, going up along the towers that actively pump that carbon dioxide into the atmosphere, ending with a top view of the overall forest plot. I thought it would be useful to show a little bit more on what I mean when I say that a cylinder copy would lead to better understanding on how much carbon is stored in these trees. So on the left-hand side, you have a bigger picture of what my cylinder copy from a particular oak tree at BIFoR FACE looks like, and a little bit of a zoom in on certain branches that are color-coded for a number of different things. And then we have an even more simplified version of some cylinders that make our example tree in this case. And I gave them some imaginary numbers on how much volume would be in these particular cylinders and the steps that I would go through to, in the end, determine how much carbon would be in this particular tree. So summing up, the 4, the 7.5 and the 6 would give me 17 cubic meters. And knowing the equation that my biomass is built up from volume times density, I can give those numbers, the 17 and a generic 750 kilograms per cubic meters as an estimation of our biomass for our example tree here. Knowing that fresh wood contains about 40% of carbon in the total weight, so that would exclude then other molecules and water, of course. A lot of fresh wood contains a lot of water, and in this case, for our example tree, that would boil down to about 5,000 kilograms of carbon in our imaginary tree. And this is the principle that I will then use for all my different point cloud copies, my cylinder copies, on all the different rings or arrays that are at BIFoR FACE and EucFACE, giving me a pretty accurate carbon number for these trees that were exposed either to ambient air or elevated CO2 air. However, a tree is not a static thing because of certain factors. Some twigs and branches will fall down over time. And that's also part of my research, is looking at what is removed from these canopies after that scan was taken and how much carbon would go into the next step in that ecosystem. So how do I do that? I do a transect inventory, which is illustrated here on the left. I placed in all the different rings a yellow line, and on a monthly basis, I walked past that line and looked if any new material fell down on my transects. And then I gave them a tag, which would help me the next month see, oh, this is already tagged material. I do not have to go and measure this again. And that gave me about a year, a little bit more worth of data on branch turnover, as we call that. This is, of course, a subsample because, again, I cannot make an inventory of the complete forest floor in all these rings. That would be a massive task. So using that subsample data, I am working on a model that will give me, well, using the information that I found on my transect, expand that to a number that reflects what fell down everywhere in those rings. I mentioned before that besides working at BIFoR FACE, I'm also working at EucFACE, which is a very similar experiment located in Sydney, Australia. At EucFACE, I'm gonna do the exact same research as I've been doing at BIFoR FACE, with one interesting additional aspect with that eucalyptus trees, which are the dominant species there, also shed bark on a regular basis. So I am alongside the inventory of twigs and branches on the forest floor. I placed bark traps along certain trees in the forest, and you now see a sped up video of me sorting through one of those also monthly inventories where I am separating the bark litter from the leaves and the twigs and the fruits that I can later use to define how much bark is lost from these trees over time. And with that, I want to recap a bit on what I've been telling you for the last few minutes about my research. So I'm using digital copies of the trees in both the treatment and the control plots at 2 phase experiments. And I made monthly inventories of all the tissue that came down over a number of months, using both models and observations to get that subsample that I've been collecting on the forest floor to a forest-wide number, and then translate that to carbon numbers in the end. And all of this together will help determine how the trees of the future will respond to elevated CO2 levels by looking at their woody tissue dynamics. And with this video, I would like to conclude the 20-minute lecture from EucFACE this time instead of BIFoR FACE. I hope you enjoyed and learned a bit more about the research I've been doing there.